Okay, so we're going to look today at protein structure and function, and I'm going to actually split this video into two parts. The reason being that I think it kind of makes sense to do the sort of fundamentals first before then looking at protein folding. So these are the objectives, so we'll make a start. The first thing we're going to look at is our GCSE recap. And what you should remember, hopefully, or if not, sort of at least pay attention to this bit a little bit more, perhaps. From GCSE, we know that proteins are examples of polymers. Okay, they are large molecules made up of repeating units of monomers. And the monomers that make up proteins, if you imagine a protein as perhaps a long chain of these monomers here, these kind of blobs, each of these individual monomers, I'm highlighting one in green here, is an amino acid. Okay, an amino acid, and we're going to look at the structure of those in a minute. And the other thing to perhaps remember is that this linear chain of amino acids actually folds up. It folds up into a three-dimensional structure with a specific shape. Okay, so a specific 3D shape. So it might then look like this. And I'm just doing a very quick sort of cartoon version of it here. That's actually not particularly helpful. Let's try that again. There we go. That's a bit better. And that may have been represented in a textbook and things as like a blob that looks like this. And hopefully that looks a bit familiar to you. I've tried to make it look a bit like an enzyme, which would be an example of a protein. And of course, you've got your active site in the middle here. And it's the specific shape of the active site and that dictates function. And that's really important theme for the next sort of topic, which is that this 3D shape dictates the function of the protein. That's really an important message here. And I guess just to point out that the other thing that you came across is the enzyme that actually breaks down proteins back into amino acids. So there's a protein that can come along, there's an enzyme that can come along, that can chew or break these bonds to liberate individual amino acids. And that we sort of refer to as a family of enzymes, and they're called proteases, or protease, it may have been described as. One of the examples of that is in the stomach, where we use the example of pepsin. Okay, it breaks down proteins into amino acids. So that's pretty much all of I think I'll leave that there in terms of GCSE. So what we're now going to do is the first A-level bit, which is zoom in and look at the structure of these amino acids. Okay, what do they look like? And they have this particular structure. The first thing is that they have an amino group, and hence the name, an amino acid. So this means an amino group, which from hopefully what you can remember from revising your functional groups, looks like this. There's a nitrogen and two hydrogens attached to it, and this can be attached to the rest of the molecule, which we just show by an R group. So that's just a carbon atom, the rest of the molecule, essentially. So that's an amino functional group. What makes this whole thing an acid? The answer is it has a carboxylic acid group attached. So again, we'll put our R group in. There's a C double bond O bond into a hydroxyl group. Okay, so this is an amino functional group, and this over here is an acidic functional group. It's a carboxylic acid functional group. Okay, so how do these join together? Well, we put a carbon in the middle first of all, and it's often referred to as the alpha carbon. Okay, that sort of symbol there, it means alpha. Don't worry too much about that, but it's the kind of carbon in the middle, and it's the one that exhibits chirality, which we will touch on at some point, but it's not particularly important for A-level biology. So connected to this carbon, we need to draw our carboxylic acid group, which I'll do in blue. There we go. The other side, we're going to actually then draw our amino group. So I'll do that in red. There we go. And then we have a hydrogen. I'll just do that in black. Fairly straightforward. And I'll swap color now to green because we then have what we call an R group. And that's the structure of a general amino acid. Okay. And I'm just going to sort of point out now briefly what this R group is. We'll touch on it later. But this R group here represents a variable group. Okay, so it could be one of a number of things. And this can change. Okay, it could be something as simple as a hydrogen atom again. It could be a methyl group, it could be an ethyl group, and it could be it could be a charged group. And it could be as complicated even as a sort of benzene ring type structure. We'll come across what some of these R groups look like later. You do not need to remember any of them, but you need to be aware of how different in terms of chemistry they are. So chemically, how they differ from each other and all the different variations that you can have. So that's the R group. We then turn our attention to the amino group over here. And it's worth pointing out that don't forget that the NH2 group has a lone pair of electrons sitting on that nitrogen. Okay, that gives nitrogen 5 electrons in its outer shell, which is what it has. And they become important later. It's also important to note that this is a base. So this chemical group acts as a base. What that means in chemistry, in sort of Bronsted-Lowry acid-base terms, it means that it's a proton acceptor. So it accepts protons, which are H+ ions, right? It's the hydrogen atom minus the electron. And hopefully you can appreciate how it's going to do that. It will use the lone pair of electrons here to make a dative covalent bond, in blue here, to our proton here, okay? The dative covalent bond, hopefully you remember from the biochemistry stuff, basically just means that the electrons that are used in that covalent bond originate solely from that nitrogen atom. Okay, it's not that hydrogen is providing 1 electron and that nitrogen is providing 1 and they're sharing those. Nitrogen is providing both electrons here. Hydrogen doesn't have any because it's a proton. So that's all we sort of mean by that. But that's essentially how it's going to do it. So I'm just going to get rid of that for now. But if you want to just learn that it's a base and it accepts protons, that's absolutely fine. Zooming over here to our carboxylic acid group, hopefully you remember that carboxylic acids are acidic. This is an acid. And in the same way, instead of accepting a proton, this is a proton donor. Okay, it releases protons. So here we have a molecule with sort of interesting chemistry. It has both a basic group within it, the amino group, NH2, and it has an acidic group, the carboxylic acid. In a similar way to how phospholipids were both hydrophilic and hydrophobic, and we call that amphipathic, there's a name for being both an acid and a base in the same molecule. Okay, and this is a word, not amphipathic. So amino acids are amphoteric. They have properties, they exhibit properties in the same molecule of both an acid and a base. In other words, they both accept and donate protons to solution. So this is important because at pH 7, at about pH 7, physiological pH, what does your amino acid actually look like? It does not actually look like this. At pH 7, our carboxylic acid group acts as an acid, and our amino group acts as a base. So in other words, our carboxylic acid group will release or donate its proton to solution, and the amino group will pick up a proton from solution. Same proton, but if you wish to learn imagine that it is, then that's fine. But so the amino acid will actually look like this. So let's put our alpha carbon in. Our carboxylic acid group will donate its proton here to solution, and this oxygen therefore becomes negatively charged. Okay, this proton is given to solution. Now, what's going to happen to that proton? Of course, you have water knocking around. And one of the lone pairs on the oxygen here can therefore form a dative covalent bond to that proton, and we then form H3O+. But don't worry too much about that. But that's what's going to happen. Essentially, the water is going to pick up a proton there. On the other side, we have our amino group. And the lone pairs here are going to even be, from water, perhaps, or if it's free floating for a bit, then it can pick it up there. So I'm going to add a proton on here, and that means that the nitrogen is positively charged. So we have two charges now. We've got a negative charge on the carboxylic acid group and a positive charge on the amino group. And if I just put my hydrogen in here and my R group, there we go. That is what, if I just get rid of this for now, there we go. That is what our amino acid looks like at pH 7. It has both a positive and a negative charge. Okay. This particular structure is called a Zwitter-ion. Okay. And all this means is we have essentially our amino acid that has both positive and negative charges on it. Now, the only thing just to add to this point is this is pH dependent. So at pH 7, I've said here, we have this particular structure, our Zwitter-ion, where the carboxylic acid group, has donated a proton, and the amino group has picked up a proton. They've acted as their acid and base. What you can hopefully appreciate is actually that this is all pH dependent, and we'll come back to this at the end of this particular video. If we decrease the pH, what this means is we are increasing the concentration of protons. Okay, there are more protons in solution if the pH is low, because if you imagine the pH being low, right? That's to say that, that's what acids are, right? That means that they're obviously donating their protons, as has happened here, into solution. The more that happens, the more protons you have floating around. So if you move an amino acid into a region of into a solution rather of fairly low pH, that means we have quite a few protons floating around. Which means, and I stress that this is, it depends what pH we're talking about here, and we'll get back to that. But generally speaking, as you decrease the pH, more and more of your amino acids are going to be protonated. As you increase the pH, oops, I don't know what happened there. Something weird with my surface. Try again. As you increase pH, what that means is you decrease the proton concentration. And if there are not many protons around, then your amino acids are going to release protons into solution to counteract the fact that you've got less protons knocking around in solution than you had before. So what this means is your amino acids kind of act like a proton buffer. When you've got lots of protons around, what's going to happen here, if you've got lots of protons around, both groups are going to be protonated. So if we look at our carboxylic acid group, if we've got lots of protons around, we're going to easily stick one onto the OH group here. And the amino group is going to pick one up as well, because there's lots of protons, here we go, around. So this is what your amino acid is going to look like at sort of low pHs. Okay, it's every possible group is going to be protonated, right? So here and here, because at low pH, protons around. The opposite is true. So at a high pH, we have not very many protons in solution, which means your amino acid groups are going to donate protons. So as you increase pH, you're more likely to form this structure where the proton has been given away to solution here. So I'm just going to get rid of that now, but the carboxylic acid group has given away its proton, becomes negatively charged. And the amino group will have done the same thing. So if I draw that here, particular structure is going to dominate at high pHs. And of course, if you are in the middle, slap bang in the middle of these two is where, roughly physiological pH 7, that is where your Zwitter ion occurs, where we have both the protonated side over here, but the deprotonated side here on the carboxylic acid, which is what we've drawn here. So it's kind of the middle ground between the two. Now, at what pH this becomes true depends on different amino acids. And we'll get to that in more detail later. But for now, just appreciate that if we alter the pH, we change the ionization states of our ionizable groups, the amino group or the carboxylic acid group. And at pH 7, it just so happens that both groups are charged. The amino group picks up a proton, becomes positively charged, and the carboxylic acid group releases a proton, becoming negatively charged. So I'm going to leave that there, apart from just to say that the R group becomes important here. And I will pick that back up when we do stuff about pH and pKa values. But it's worth bearing in mind now. So that's the structure of an amino acid. That's what a Zwitter ion is. And actually, your amino acids in your cells exist absolutely in this form, the Zwitter ionic form, the doubly charged version. So that is our amino acid structure. So we're now going to look at a condensation reaction. So as with everything, how do we make the polymer? Okay, so it's all well and good having our individual amino acids. So I'm going to draw one here in black. There we go. And I'm going to draw one in blue. Now, for the sake of argument, bear in mind what we've just said about Zwitter ions. I'm not going to draw the Zwitter ionic forms. I'm just going to draw the standard forms like this. This is what you will be expected to draw in the exam unless stated otherwise. So unless you're told to draw the Zwitter ion, you can just draw them like this. And actually, it makes life easier when talking about how these things react to join together. Okay, so let's look at these two monomers. We've got two amino acids, and they are going to join together, okay, because hopefully what you can appreciate is we've got oxygens and hydrogens close to each other again. Okay, so what's going to happen is the OH group here is going to bond with one of the hydrogens on the nitrogen, and that is going to be liberated as a molecule of water. There we go. I've tried to color code it there for you. So this is a condensation reaction. There we go. And of course, water is liberated, right? Not always, but in most examples we'll come across, it is. And what we're left with is a bond that now forms, if I just do it in green here, between this carbon here and this nitrogen here. So let's have a look. We can now draw the dipeptide that's formed, the two amino acids. So starting with the black one here. That's that. I'm going to draw the bond in red. Actually, I may as well stick it in green. There we go. To the nitrogen, bonded to the hydrogen. Alpha carbon. And our carboxylic acid group. And there you have it. We have now joined together two amino acids. Okay, so this in black here was, oops, that's not very helpful, was amino acid number 1. And the amino acid in blue here was amino acid number 2. The bond that we formed in green here is a special type of bond that you need to know. Now, the chemists amongst you hopefully recognize that this whole here is an amide bond or an amide linkage. However, in biology, we don't tend to call it the amide bond. We give it its own special name, kind of like a glycosidic bond for carbohydrates. We call it a peptide bond. Because actually it's really important with how when we formed a polymer of all of these amino acids to form a protein, of course, this peptide bond is really important. And actually what we often call or refer to proteins, there's a caveat to this, but generally speaking, we refer to proteins as polypeptides. Okay, so once we've linked, we can add another amino acid, by the way, to here, and we can keep going. So essentially, we've put two together. Now we can add another one on. We can add another one on, and this then eventually folds into our 3D shape. And we actually call this very often a polypeptide. And hopefully that term makes sense because we're going to have lots of peptide bonds, right? That's a peptide bond. That's a peptide bond. Okay, and we can put hundreds of these together. So that's the condensation reaction that happens. That's what you sort of need to be able to draw. In terms of a mechanism, how this works, hopefully you should be able to sort of do this, or at least sort of come up with a vague sort of appreciation for this already. So let's again draw my amino acid here. Oops. There we go. Let's do the well. Keep the same colours. Let's do one in blue. There we go. So, how does this work? Well, I'll swap to purple for the mechanism. As I mentioned, there's a lone pair on this nitrogen, right? Nitrogen forms 5 bonds. It's only forming 3. It therefore has nitrogen has 5 electrons rather in its outer shell. It's using 3 of them in bonding, so it has 2 remaining. These act, or this whole group here acts as a nucleophile, which we've come across. Okay, there's a lone pair of electrons here. That's a lone pair, remember, that is capable of forming bonds to electro positive or electron deficient groups. And if we look over here on the carboxylic acid group, what you can appreciate is we've got lots of oxygens. That was a bit bigger than I thought. There we go. So this oxygen here, is going to be delta negative. This oxygen here is going to be delta negative. And what that means is the carbon in the middle here is going to be delta positive. So what can happen is, and if I just get rid of these delta negatives, right, the carbon group here is delta positive. So what's going to happen is these electrons are going to attack that delta positive carbon because it's electron deficient. It's an electrophile. I'll write that down. And what's going to happen is then, I'm going to simplify this slightly. What then happens essentially is the 2 electrons in this covalent bond here are both given back to the oxygen. Remember, double-headed arrow to mean both electrons are being transferred back to that oxygen there. So what we then end up with is this. So again, I'll get sick of drawing this by the end. We produce OH minus because that oxygen has gained an extra electron that it didn't have. Remember, let's say this cross electron here, this one, actually used to belong to this carbon in a covalent bond. It now belongs to the oxygen. Okay, so it's actually got an additional electron there, making it negatively charged. So that's our hydroxide anion that's produced, OH minus. And then we formed the bond. I'm going to do it in purple here to reflect that it's these electrons here forming the bond. So that then forms a covalent bond. I'll highlight it in yellow as well, just to make the point. There we go. And the 2 electrons are here, of course, to the nitrogen, which currently has 2 hydrogens attached. We'll get back to that in a minute. Then we've got the alpha carbon, R group, and our carboxylic acid group at the end. So the only problem here, of course, is that the nitrogen is forming 4 bonds. So what then happens is our hydroxide group, the electrons there, will pick up this hydrogen. The reason it can do that is because of the delta charges again. This nitrogen, if I do it in green, is delta negative, and that makes these hydrogens both delta positive. So the oxygen can pick up this hydrogen, and the 2 electrons in this covalent bond then get given back to the nitrogen to reconstitute its lone pair. And what we then end up with is what we drew in the previous picture, so up here. Okay. So essentially what happens is this proton then disappears. There we go. Off it goes. And that's essentially what we've got. And then this OH group picks it up, becoming water. And we then have our peptide bond that is formed. I'll point out now that this process happens again and again, but we add amino acids, okay, to this end, right? The next amino acid comes in and connects to the carboxylic acid group, okay? We need an amino group, of course, on the next amino acid. And then we can do the same thing, right? Where we have nucleophilic attack, carboxylic acid end. Okay, we don't add to the amino end. So what that means is our molecule is sort of polar in a sense. It has directionality to it. On one end, the sort of first amino acid that went in has the amino group sticking out the end. So it's called the amino terminus. Okay, or it's referred to in sort of biochemistry lingo as the N terminus. At the other end of the protein or the polypeptide, bearing in mind we could have hundreds of amino acids stuck together, we have our carboxylic acid group sticking out. So this is called the carboxyl terminus or the C terminus. Okay, so if you were going to look at a long chain of amino acids, and I'm just going to stick them together like this, imagine you've got hundreds of them. On the first amino acid that we put in, we will have an amino group sticking out the end. And on this one, we would have a carboxylic acid sticking out the end. Okay, so you can identify and the last amino acid to go in was this one. Okay, so the protein's got direction, right? It's not symmetrical. The two ends look different. So that's the condensation reaction. Just to finish that off, I just want to talk about the peptide bond very briefly. And then we're going to look at some Zwitter ions, just to make the point. So let's just look at the peptide bond. The peptide bond has the following features, and they're really important. So if I draw the peptide bond like this, it looks like that. And of course, it connects either side to the rest of the molecule. I'm just focusing in on the peptide bond. Hopefully, your brain is already thinking, ooh, oxygen, delta negative. And is also thinking nitrogen is delta negative. That means this hydrogen must be delta positive, right? Because of the electronegativities, right? Remember, the electronegative atoms we come across are Fon, F-O-N. And hopefully what you appreciate that because we've got hydrogen attached to this nitrogen and it's delta positive, this is absolutely used for hydrogen bonding. Okay, it's got hydrogen bonding potential. So that's the first thing to say. And of course, this electronegative oxygen does as well if it's close to another hydrogen that's delta positive. But we'll come back to that later on anyway. So I'm just going to get rid of that for now. Do keep it in your own notes. But what I want to sort of point out is that the peptide bond actually isn't free to rotate. Single bonds are free to rotate around. Okay, so this molecule sort of exists in two halves that can kind of rotate with respect to each other. Okay, so essentially this hydrogen could move up here, for example. The whole thing could be flipped. That actually can't happen. And the peptide bond is actually stronger than we would expect. It's more difficult, harder to break. Okay, it's more stable than we thought. And also it can't rotate. It's not free to rotate. Okay, so it has a sort of restriction placed upon it in terms of what conformations it can take on. Which is weird because single bonds have free rotation. The reason is because of something called resonance. Oops, my pen has stopped working. 2 seconds. Hopefully we're back. Resonance of the peptide bond. Okay, so we have this thing called resonance, and this works because the nitrogen here has its lone pair of electrons. And what can happen is obviously we've got our delta negative here, delta positive, as I talked about before. The lone pair of electrons can, this nucleophile is very close to the electrophilic carbon, delta positive. So what it can do is form a bond with the delta positive carbon here. So that's really bad arrow, apologies. There we go. And what then happens is I'm trying to swap color here. Here we go. The electrons in one of these single bonds on the oxygen then break. They get transferred back to the oxygen. And so what you end up with is this structure, where we have a carbon here with a single bond oxygen. We've formed the additional green bond here to the nitrogen. So it's now a double bond to the nitrogen, and obviously both sides link on to the rest of the molecule. Okay. Structures exist in proteins, and these exist in sort of an equilibrium with each other, is what I'm trying to say. And the reality is, the truth is that the true form exists somewhere in the middle. So the peptide bond exhibits kind of both properties, right? Essentially, the peptide bond here that we're really sort of focusing on isn't just a single bond. It can sometimes be a double bond. And in truth, it exists as a sort of 50-50 mix between the two. And it's called resonance, that property. So we say that the peptide bond has partial double bond character. And all you need to really be worried about is that this gives it increased stability, but it means we cannot rotate. So we can't rotate a double bond, essentially. So it's not free to move. It's relatively inflexible and can only take on certain conformational shapes. That's all I'm going to say about that, but it's an interesting point just to make you aware of. So, just before I'll tick it off first. So condensation reactions done. So what I now want to do is just look at the R groups, and we're going to look at these Vita ions as well and recap that. So I've got an image over here, which shows pH down the bottom here, and we've got concentration on the y-axis over there. And you can see the Vita ion here is what exists at pH 7. Now, so pH 7 sort of around about here. Okay, the Zvita ionic form dominates, and actually it dominates across a range of pHs, right? From about pH, probably from about pH 3, pH 4 certainly, to about pH 8 or ish. You have your Zvita ionic form. Ionic form, which is where we have, okay, both the deprotonated form on the carboxylic acid and the protonated amino group. Okay. Now, as we decrease the pH, this increases, of course, the number or the concentration of protons in the solution. And that's shown here. So what that means is, as you decrease the pH away from sort of pH 6, pH 7, you start to increase the concentration of protons, and therefore both groups are protonated. So you start to increase the sort of percentage of molecules, percentage of amino acids that now have a proton on the carboxylic acid group and have a proton on the amino group. This amino acid, in this case, would have a net charge. It would be positively charged at low pHs, which hopefully you can appreciate is important for protein structure. and we'll see why later. At pH 7, we've got our Zwitter ion. It is neutral. Oops. The reason it's neutral is because we have both a positive charge on the amino terminus and we have a negative charge on the carboxyl terminus. They cancel each other out. The caveat to that is coming. As we increase the pH, we decrease the concentration of protons. And your amino acids, in a way, act like a proton buffer. So what they then do is sort of release protons into solution. So what that means is as you increase the pH here above pH 10 or so, increases. So more of your molecules are in the deprotonated form. So your carboxylic acid group will not have its proton. It's negatively charged as a result. But your amino group hasn't picked up a proton. It is also deprotonated. Okay, to sort of increase the number of protons in solution. Okay, it comes back to Le Chatelier, actually, from GCSE chemistry, right? Look that up if you're interested, or email me. So this guy dominates at high pHs. Okay, now the proportion of molecules that exist in either of these three forms depends kind of on the amino acid we're talking about and the exact pH that we're talking about. It's not a binary switch. It's a gradual sort of change. Okay, and we can predict that, which we'll come to next. Okay, but it's just worth showing you that. So the only thing I want to just point out is we form an acid group over here and an amino group over here, and that they are in this form. In other words, not Zwitter ions. And hopefully what you're asking yourself is, if we have our Zwitter ion as the most common form in cells at pH 7, like that, how can the reaction still happen? The nucleophilic attack here that I said happened like this, how can that still happen? And the answer is it still does. And I'm not going to give you the detail on that. You don't need it, and it will make the video too long. But just suffice to say, if you were going to draw it like this, you can simply take the oxygen here, take 2 of the protons like that, and that produces your water. And what you hopefully appreciate is you actually end up with the same result, right? Your peptide bond would be from this nitrogen to this carbon, and it looks the same. Okay, so it doesn't matter that these are charged potentially. The same reaction happens. Just worth making that point in case it's confused you. So what we're now going to look at is the side chains. Here we go. So these are the R groups, okay? And all you need to be aware of, you don't have to learn them. There is one that I think you probably should learn, certainly, but we'll get to that later. The R groups, you need to be able to point out, okay? So look, alanine, for example. How do you identify the R groups if they're not highlighted? Okay, so I'll make this point up here. How do you identify the R groups? Number 1 is circle your amino group. Second step is to circle your carboxylic acid group. Third step, circle your hydrogen. Oops. And the fourth step is whatever's left is the R group. Okay, so just very quickly, if I gave you an amino acid that looked like this, like that. What you need to do is circle the amino group here, circle the carboxylic acid group. And by the way, I've deliberately tried to trick you here. I've put a carboxylic acid group on the bottom of this R group here. Start with the amino group. Once you've got the amino group, look for your alpha carbon. It will be attached to it. It'll be the next one. And the carboxylic acid group that's directly attached to that is your carboxylic acid group of the amino acid. Then circle your hydrogen, and then whatever's left is your R group. So actually all of this, run out of colors, let's do it in purple. All of this is your R group. Okay, so that's how you do it. Okay, so start with your amino group, then look for your alpha carbon, look for the carboxylic acid group that's attached to that, look for your individual hydrogen, and then whatever's left is your R group. Don't be confused. R groups can have carboxylic acid or amino groups on them. So just don't, just be careful there. Here, however, they've highlighted them in purple. Okay. So alanine, for example, we would look for the amino group. Here it is. Oops, shouldn't use that color really. Here we go. Look for the amino group. Look for the alpha carbon. Circle the carboxylic acid group attached to it. So here we go. Look for the hydrogen, and whatever's left is your R group. So there we go, CH3. And you can see that we've got something called aliphatic R groups. Don't worry about that. It just means it's non-aromatic. Okay, so it's not like a benzene. Right, you can simply have a hydrogen on glycine. You can have a methyl group for alanine. Proline has this weird kind of V-shaped structure here. So that's quite important for proteins. Valine there, you've got leucine, which has got more sort of hydrocarbons. Isoleucine, a sulfur group in it. Interesting. Hopefully you can appreciate, though, that the backbone of those amino acids is all the same. Very quickly, you can have uncharged polar groups. So typically with OH groups or double bond oxygens. So serine, for example, it's CH2, there's an hydroxyl group. So delta negatives, lone pairs, think about that. You've got a sulfanhydryl group here, which is important for cysteine, and that's the one that I would actually tell you to remember. Okay, remember it's got this SH group. It's got a sort of thiol group is the sort of slang term. So you can read through that in your own time. You then move on to charged groups. So you can have negatively charged R groups. So in this case, some of the R groups, look, have carboxylic acid groups. But don't be fooled. If this wasn't color-coded, how would you know that that's actually part of the R group? Well, you would circle your amino group. Here we go. Look for the carbon that's attached to it. There it is. Look for the hydrogen and the carboxylic acid group that's directly attached to it. So the hydrogen is here, the carboxylic acid group straight away. Everything else is your R group. Don't be fooled that there's a carboxylic acid group there. Same thing with glutamate there. You can have positively charged ones. Again, you have NH2 groups typically here. So NH2, look, or NH3 in this case. So you can have positively charged amino acids. Again, don't start labeling it here, right? Look for an amino group attached to your sort of carbon that is attached to a carboxylic acid group, right? And you can have your aromats. Okay, so something like phenylalanine looks a bit like a benzene ring there. In fact, it is. And tyrosine, tryptophan. Hopefully, you can appreciate there are 20 amino acids here, and they all have different chemical properties. And the chemical properties that they exhibit determines or will help determine protein function. You don't need to learn the R groups. You need to be able to identify them from a picture, from a sort of skeletal diagram or a displayed formulae sort of representation, but you don't need to learn them off by heart. The only one I would recommend you learn, you'll see why later in the next lesson, is cysteine, because it has this thiol group, SH group in it, which is important. So that is that. So I'm going to tick off R groups. Make sure I get the right color. There we go. So R groups done. And the final thing we're going to look at is Ka values and pKa values. I'm going to not make too much of a song and dance about this, but it is important to know. So we're going to look at pH and something called, we're going to look at Ka values and pKa values, which are essentially the same or a very similar sort of thing. But we're going to look at those now. So it's important to know, and this is the key message with all of this. We said that the ionization state of ionizable groups in your amino acids, okay, depends on pH. Okay. In other words, whether you're protonated or deprotonated, that's what we kind of mean by this. Do we have a proton? So protonated or not. Whether we are protonated or not depends on pH. And I alluded to that earlier, or I sort of explained that earlier, because of course, pH is a measure of concentration of protons. And if you're interested, the equation for pH is taking the minus logarithm to the base 10 of your concentration of protons. Okay, and that, of course, is on a scale from 1 to 14, whereby pH 1 means there are a high concentration of protons. And if you're up the other end, pH 14, there's a very low concentration of protons. Okay, the reason why we minus log this number is to give us numbers that fit into the pH scale, right? 1 to 14. Otherwise, the absolute value of this proton concentration, is just a sort of number that's really hard to get your head around, right? It would be, 1.7 times 10 to the minus 8, a sort of really difficult number to work with. So that's the only reason why we minus log that number. It gives it turns it into a sort of workable number on the scale of 1 to 14, of course. So we've talked about ionizable groups, and essentially what these things allow us to do, Ka values and pKa values, is we can predict whether we're going to be protonated or not. Okay, so pKa and Ka values can allow us to predict if the group, the chemical group we're looking at, the ionizable group, will be protonated or not. Okay. And that's important in biology because we don't always work at pH 7. I mean, we obviously do in cells. pHs, right? How pH impacts on enzyme function, for example, is it alters the number of protons and it changes the ionization state of our groups. The only other thing to mention before we sort of get cracking with this is that by ionizable groups, we of course have come across this already. In your amino acid, you've got 2 ionizable groups that I mentioned. Right? You've got your carboxylic acid group and your amino group. They're both ionizable. The amino group can either pick up a proton or not, and the carboxylic acid group can donate a proton or not. However, this is where our R groups come in. Hopefully, some of you have just thought, hang on a minute, didn't we just say that the R groups can be charged? And the answer is yes. So actually, first things first, we don't really bother with whether our amino group or our carboxylic acid group are going to be protonated or not. Because as we said right at the top somewhere, I hope. Or was it over here? It was over here. As we said over here, even if you're charged, so here and here, the same reaction happens to form the peptide bond. Okay, we will still end up in this situation regardless. So actually, do we really care whether these groups are protonated or not? Because we would if the amino acids existed individually, but they bond together to form peptide bonds, right? This bond is together, and so we lose the charges. So actually, whether these groups are charged or not doesn't matter when we're looking at proteins. Okay, because they're going to be the amino group and the carboxylic acid group are going to be tied up in proteins. Okay, so that's what I'm trying to make a point of here. We don't really bother with whether those are ionized or not. We absolutely need to bother with the R group down here. So I'll do this one in green, right? Absolutely need to bother and decide whether that is going to be charged or not. And you'll see the importance of that when we look at protein folding in the next lesson. But essentially, it's the R group ionization state that we care about. And of course, some of the R groups are ionizable. So just to put that into notes, the carboxyl terminus and the amino terminus don't really matter too much. Because whether they are charged or not doesn't matter, because they are used regardless to make the peptide bond. And when they've done that, they don't have a charge anyway. So even if they were charged before, the Zwitter ion, they don't retain those charges once they've produced the peptide bond. We care very much, so I'll put here but, R groups can be ionizable, right? What we mean by that is the R groups that give each amino acid its own sort of character, if you like. Some of them can be positively charged, right? We came across those. So, R groups can have carboxylic acid groups and amino groups, so that become NH3 plus. This becomes O minus, right? And of course, these aren't tied up in making a peptide bond, so they are free. They're kind of exposed, if you like, in the protein. And therefore, the charge they have matters a great deal, right? It matters whether this guy in a particular amino acid, R group, is negative or not, or whether it's got a proton and it's not negative. It matters whether the NH2 group has picked up a proton and become positive or not. So that's the sort of background. Linking on to Kas now. Okay, so how do we know this? How can we tell? How can we know whether the R group in question is going to be charged or not at a given pH? Well, think in terms. Is true, by the way, for your amino group. Okay, but I'm going to focus on the carboxylic acid because it's easier to sort of explain initially. So let's take this group. It could be like this. Okay, it could be in its protonated form, or it could be in its deprotonated form. Okay. And where the equilibrium between these two lies determines or is determined by how strong the acid is or not. Okay, this is an acidic group, and if it's a strong acid, okay, which it isn't, but just to make the point, strong acids, they completely dissociate in water. Okay, so we typically refer something like hydrochloric acid will completely dissociate. The equilibrium is so far to the right that we actually, it would sort of look like that. We actually don't really bother even writing the equilibrium to say that it completely splits up in water, right? Very quickly, and we assume all hydrochloric acids sort of molecule up. However, carboxylic acid groups here are actually weak acids, so we can't assume this. Okay, so this isn't actually true. What's true is what I've drawn here, which is where we have an equilibrium, and whether that that equilibrium is massively favoring the deprotonated form or whether it's massively favoring the protonated form depends. Okay, so just to add to this, we represent the acid as HA because it could be any kind of acid, not just hydrochloric acid, and it could be the COO, the carboxylic acid group. This will dissociate with an equilibrium to form H plus and Cl minus, A minus, right? Whatever that may be. Okay, this is a weak acid. This is what a weak acid does, but we don't know where the equilibrium lies. If it lies further to the right, then it's a stronger acid. But if it lies further to the left, it's a weaker acid, right? Not much of it is dissociating. Concentrations. And all you have to do is measure the concentration of the undissociated your protonated form of the acid, and you measure the concentrations of the unprotonated form of the acid and the number of protons that are released. Okay, measure concentrations. And we could put this into a mathematical equation. We could say that if we take the concentration of the products, the H plus on its own and the dissociated form of the acid, and we divide it by the concentration that's left of the undissociated acid, this gives us a fraction. And we call this the acid dissociation constant, or Ka. Okay, so the acid dissociation constant. And if you imagine this as a fraction, if Ka is large, it's a big value, that means these guys up here must be quite large numbers, and the concentration of the undissociated form must be low. So what this means is if Ka is a big number, then we can say that the concentration of free protons is very high, and the concentration of the protonated form of the acid is very low. So essentially what this means is it's a stronger acid, okay? Lots of the molecules, lots of the acid exist in the dissociated form, deprotonated form. Okay, so if this was carboxylic acid and it had a large Ka value, it would most likely, most molecules would look like this, right? It would have kicked off the proton. So, just to be clear, if I was going to put carboxylic acid into this equation, the concentration of the proton would be the concentration of the proton. Concentration of A minus refers to the conjugate base, so the deprotonated form of the acid. And of course, HA is the protonated form, so this represents that form there. Okay. And the reverse is true if it's a small number. So if Ka is a small number, then we have lots of the protonated form. Okay, so the carboxylic acid, if the Ka rather was small, would look like this. Okay, hopefully that's clear to you. However, just to confuse things a little bit more, we don't tend to use Ka values in biology because they can be weird numbers. So in the same way that we convert proton concentration to pH, right? I take the Ka and minus log to the base 10 of it, like that. This gives me not the Ka, but the pKa. It's kind of like the pH earlier. And this just gives us easier numbers to work with. We can work with this number a lot easier than we can just the Ka directly. Okay. But the thing just to point out is we have minus logged it. So what that means is the opposite is true for pKa as it was for Ka. So in this case, if pKa is small, if it's a small number, that means it's a stronger acid. Okay, that means we have a lot of the deprotonated acid. If the pKa is large, that means we tend to have more of the protonated form of the acid. Okay. And you can look up these values for different things, right? The important point here is that different R groups, so different ionizable R groups, variable groups, have different pKa values. Okay, and it's important to note that the pKa value is a constant. Okay, so this does not change. Each R group has its own distinct pKa value. Okay, so the next thing to point out is I'm going to sort of pose a question to you. There's a special relationship between pH and pKa, obviously, or I hope it's obvious. What if the pH equals the pKa? What does that mean about our ionizable R groups and our molecules that we're looking at? If we have an R group that, I don't know, has a pKa of 6.3, and we put it into a solution that has a pH of 6.3, what does that mean? Well, this bit, by the way, is kind of extra, so don't worry too much about this, apart from the equation that we get to at the end, but we'll get to that. So this is kind of a little bit of maths to prove this, but it's quite nice if you follow along. So we know that pH, if it equals pKa, we want to know what is the concentration of the deprotonated form versus the concentration of the protonated. Are we going to have more protonated amino acid R groups, or are we going to have more deprotonated amino acid R groups? The answer is this. Well, actually, if we change these to what we know they are, so we know that pH is actually the minus log of our proton concentration. There we go. Concentration denoted by square brackets. And the pKa is the minus log of our dissociated acid and our protons, concentrations, over the protonated form of the acid. So that's kind of what this means. We can now get rid of the minus log, right? It's on both sides of the equation. We can get rid of it. So what we can now do is say that the proton concentration is equal to the proton concentration and the of the protonated form of the acid. And we can now divide and therefore get rid of our H pluses because we can divide by H plus on both sides. And that simply leaves us with 1, okay, H plus divided by H plus, right, is 1, equals A minus, the concentration of the dissociated form over the concentration of the undissociated form. And what this tells us is that if the pH was equal to the pKa, so if the pH equals pKa for the particular R group we're looking at, then we have equal concentrations of both the protonated form and the deprotonated form. Okay, 50% of our molecules will be protonated at any one time, and 50% will be deprotonated. Okay, it's 50-50. So, how does this actually link to sort of what I'm talking about? Well, I'm not going to use an R group as an example. I'm going to sort of keep it a sort of simple, and in your notes you'll find that I've used alanine as an example. And actually, I think I've got the image for you somewhere. I may as well just zoom in on this while we're here. This is just showing that we've got all our different amino acids here. and I'm giving you the R groups of serine, threonine, cysteine, etc. And it gives you this table, pKa values of the carboxylic acid group and the amino group. But we don't care about these because they're going to be used up in a peptide bond. So we're not really fussed about whether they're charged or not, but you can predict it. What we're really bothered about is this group here, right? The R group. What's the pKa values of the R groups? Okay. And we'll get back to that later, perhaps, when we do enzymes. But the one to remember is histidine. And hopefully what you can appreciate is the R group of the R group, the pKa value of the R group for histidine is 6. Very, very close to pH 7, which is physiological pH. Okay. The importance of that we'll come back to. Point is, you can look up pKa values, right? We can calculate them. Did I? Oh, I probably didn't use it. I'll draw it out if I can remember the values. Right. Let's look at how this works in practice then. How can you predict the protonation state using pKa? So predicting protonation state of R groups, or in this case, carboxyl and amino groups, using pKa values and pH values. Okay, so let's look at alanine as an example. So I will draw the amino acid alanine. It looks like this, and I'll draw it here in its non-Zwitter ionic form. So let's look at, in fact, actually, the R group isn't important, but it's a methyl group in case you're interested. So it's just CH3 here. There's no ionizable group in the R group, but let's just look at the carboxylic acid group and the amino group. The pKa value for the carboxylic acid group, if you were to look it up, is 9.1. No, it won't be that, will it? It'll be 2.3. The other one's 9.1. So the amino group pKa is 9.1. Okay. So if we are at a low pH, well, okay, I'll give you the rules first, actually. So the rules are that if the pH is less than the pKa value that we're looking at, then we absolutely are in our protonated form. That dominates. Okay. If the pH is bigger or larger than the pKa value, then the unprotonated form dominates. Okay. So let's have a look at this. Let's imagine that we're at pH 2. Okay. So here we go. Let's imagine we're pH 2. Well, first of all, actually, let's say we're at pH 2.3. And hopefully what you appreciate is because the pH equals the pKa for the carboxylic acid group, right, 2.3 here, the carboxylic acid groups, 50% of them are going to be dissociated, 50% of them are not. If the pH is 1, and if the pH is less than the pKa, which we've said here, okay, what that means is the protonated form dominates. Okay, so what that means is that we're going to be protonated on this carboxylic acid group. Okay, if the pH is 1.5, if it's less than 2.3, we're going to be protonated. If we increase the pH of the solution to, I don't know, let's say pH 4, The pH is now greater than the pKa. So we're going to be in the deprotonated form. Okay. Zooming over here, if we are at a pH that's less than the pKa, okay, so if the pH is smaller than the pKa, we're protonated, and if it's larger than the pKa, then we're unprotonated. So let's look at pH 7 as an example. Let's imagine we're at pH 7. What does the amino acid look like? Well, if we're pH 7, we have a pKa value that is smaller than, the pH is smaller than the pKa value of the amino group. And as I've said here, if we're smaller than the pKa, the protonated form dominates. Okay, so that means the amino group, if we're at pH 7, for this particular amino acid, we will have a proton here and will be positively charged. But pH 7 is far greater than the pKa value of the carboxylic acid group. So the opposite is true, okay? This will be in its deprotonated form. So actually we won't have, ooh dear, where's all that gone? Here we go. This proton here won't exist, and it will be a proton in solution, and the oxygen here will have a negative charge. Okay, so that's why the Zwitter ion form, sort of dominates at pH 7. Okay, if we were at pH 1, I just want to make it clear. Imagine we're at pH 1.5. At pH 1.5, what's going to happen to the amino group? Okay, well, the pH is far, far less than 9.1, so we're going to be protonated. Okay. However, the carboxylic acid group over here, 1.5 is far less than 2.3, the pKa value for the carboxylic acid group. Therefore, it's going to be in its protonated form. So at pH 1.5, it actually would look like this, where you'd have a hydroxyl group attached on the carboxylic acid group, and the proton would be on the amino group as well. And this links back to what I said before. Remember what I said, low pH, you're more likely to be protonated on both groups than you are as if you're a higher pH. Okay, so I hope that makes sense. It's in the notes, by the way, when you fill it out. It's all in kind of, I've sort of written it out probably in less garbled form than I've done here. But I hope that vaguely makes sense, right? The key rules are here. If you look at your pKa value for your R group, let's say, right? I've used these as examples, but don't forget, it doesn't matter really. What matters is the R group, if that was a carboxylic acid group. If your pH is less than your pKa for the R group, you're protonated, more likely than not. If your pH is greater than the pKa, then you'll be most likely more unprotonated. Okay. So, why is that important? Well, it's important for R groups and it's important for protein structure. Okay. So, why am I bothering telling you about this? Well, the answer is if we look at a protein active site, imagine it's an enzyme, right? So I'm going to draw my enzyme like this. Imagine we've got an active site that looks like that. And on one amino acid here, we've got an R group sticking out, and we've got an NH3 plus group. And here, okay, we've got a carboxylic acid group. Like that. Okay, and let's imagine it's a dissociated carboxylic acid group. And what you therefore have is an ionic bond here between the two that's allowing the active site to, have its shape. Okay, this might be at pH 7. Now, let's imagine the pKa value for the positively charged amino acid is 10.5, something like that, 10.53. .65, something like that. Okay. How does pH alter enzyme activity or sort of protein function? Well, the answer is if we were to hypothetically decrease the pH, this increases the proton concentration. Okay. And as the pH decreases from pH 7, okay, we're going to encroach on. So pH decreases, it's going to get, to pH 6, pH 5, 4, 3. Eventually, it's going to encroach on our pKa value for our carboxylic acid. So the pH decreases below 3.65. When it's at 3.65, the groups, of course, are going to be protonated, right? 50-50, remember. But as we get below 3.65, more and more, the majority of the COO minus groups will become protonated. And what happens when this becomes protonated is that you no longer have a negative charge, right? We're going to draw the proton in here now. So this is now, if we were at pH, what pH did I say? Oh, well, just decrease it below 3.65. So let's imagine we're pH 2 now. What happens is we now have this proton here that wasn't there before. What that stops is the negative, or it neutralizes the negative charge that was there, meaning that we can no longer form an ionic bond between these two groups. Okay, which means that these are no longer interacting, and the active site may then look a bit more flat like that, right? It loses its shape, and we call this denaturing. And we'll come back to this with enzymes, so don't worry. I'm just making the point. Why does pH denature enzymes? It's because it interferes with protonation states of your R groups that are interacting with each other to hold the structure together. Now, what would happen if we increase the pH above 10.53? Of course, the amino group would become deprotonated, and that would, again, stop the ionic bond, right? Hopefully that makes sense. I'm not going to labor it too much. You can watch that back if you're not sure. Speak to me with any issues on that. So the only thing I just want to do finally is just talk about the pH equation. Don't worry too much about this, but hopefully the kind of the inquisitive amongst you must be thinking, what's the relationship between pKa and pH, right? Because they both involve protons. So what's the actual relationship? And the answer, I'm not going to sort of derive the equation for you just for time purposes, but what we end up with is a equation called the Henderson-Hasselbalch equation, which is that the pH equals the pKa plus log to the base 10 of your dissociated form of your acid over your associated sort of protonated form. Okay, this equation links your Ka to pH, and actually it allows you, if you know the pKa value and of your particular acid, you know the concentrations here of your dissociated and undissociated form, you can calculate the pH, okay, of the solution. And there's a question in the notes that I'd like to have a go at, please, where you simply need to plug in the numbers for pKa and the concentration of your dissociated acid and your undissociated acid, and just simply add the logarithm of that to the pKa, and it gives you your pH. Okay, I'm going to stop there. If there are any questions, let me know. That's kind of the most difficult bit done in terms of proteins. What we're now going to look at in part 2 is how proteins fold, and that won't take us long, so don't worry about that. This is the sort of more difficult stuff to get your head around. Okay, thanks very much, and do email me with questions if you have any.